with the conditions. Could I ask, is anybody happy to share? Anybody here for their first conference? Yeah, lots of hands. Fantastic. Oh, that's really good. Because that means there are some people in the audience who won't have seen quite a lot of these slides before, hopefully, which is always good. I've tried to update a little from when I've given this similar talk before, but which is the best place to stand? Is that okay? Am I in anybody's way? So, Costello and CFC syndromes are disorders of the RAS/MAP kinase pathway. And you're lucky because you're not medical, you won't get the full half hour on what the RAS/MAP kinase pathway does, which is about as much as I can talk about it, but there are people who've dedicated their life's work to understanding it. And the more we go on, the more we realise it's complex, and there are lots of things that we don't know yet. But if we think about what the clinical features of these conditions are, often the first clue that there might be a difference is when babies are bigger than expected, perhaps with extra fluid around them in the pregnancy. Sometimes the nuchal measurement at the back of the neck in early pregnancy is enlarged. And then there might be heart differences that are picked up either on the scan antenatally or when the baby's born with a heart murmur or signs that there's a heart difference. And as many of you will know, extremely difficult feeding is something that is very, very common in the early years, particularly consistent in Costello syndrome. I think one of the messages that keeps coming up is that actually Costello syndrome is a little bit more tight as a group of patients who often will have this. CFC, it's more variable, and we do have patients who've not had any feeding problems, but that's relatively rare. And shorter than average stature is very common, and learning difficulties, again, are something that we would tend to expect with Costello syndrome and CFC syndrome. And here are some pictures. Some of you might recognise yourselves or family members here. Some of these are people that we know from here in the UK in the group. Others are photographs of patients from elsewhere who've been published in the literature. And many of these photos are courtesy of Bronwyn Kerr, who many of you will know extremely well, who is the go-to person on Costello syndrome. She'll hopefully be along later this afternoon to say hello. So a bit of a genetics 101, and quite why this projects in nearly black and white, I'm not sure. That's not a good choice, is it? But our DNA is the body's instruction manuals, and the chapters of these are the genes, and the genes themselves need to be processed through this stuff called RNA, and the protein is what actually we're made up of, and the building blocks of the cells. And so for Costello syndrome, we know that the DNA bit that's affected is the HRAS gene. There's a change in that gene, usually at the 12th codon, that means that the protein that's produced works in a different way.
Just click on that.
Oh, right, okay.
Can't you see this?
Right. So we've got more than 20,000 pairs of genes in the human genome, and we get them in chromosomes, one of each pair of chromosomes from our parents. There are 23 pairs. And when we've got changes in the DNA, this can make the proteins work differently or not work at all. And Costello syndrome, CFC syndrome, and Noonan syndrome, which, does anybody in the room have a previous kind of query as to whether the Noonan syndrome might be something that can be thinking about? I suspect it's quite likely. Noonan syndrome is a very common condition compared to Costello and CFC. And what ties all of these together is that the gene changes actually make the protein do something extra. It's doing something special and gearing up the signalling through the RAS/MAP kinase pathway. But we're still learning a lot about how the different proteins work together in these various cellular pathways. And pathway suggests a sort of linearity and a predictability to it, whereas actually it's more like a network where all sorts of things are intrinsically dependent upon one another, with lots of feedback loops, feed-forward loops, and all sorts of complicated ways of regulating it that we're only starting to uncover. But within this, kinases are proteins that act as distinct steps within the pathway. And so you can get a sort of domino effect, or you can think about it as parts of an engine or a transmission, that there might be part of it that's spinning too fast and transmitting that spinning too fast onto the next bit. You might be able to tell I'm not an engineer, sorry about that for all the engineers in the audience. But kinases don't work by themselves, and there are lots of other proteins that control how they work. So they're being regulated by a whole load of other things that are things like we could call them scaffolding proteins or chaperone proteins that are actually intrinsic to how the whole system works. And here we've got a little diagram that is how it looked a few years ago. Every year or so there's a new gene or two that get discovered as a cause of Noonan syndrome in particular, and this pathway goes from looking horribly complicated to looking even more horribly complicated. But essentially, what you can see is that Costello syndrome is due to changes in HRAS, CFC syndrome is due to changes in a number of different genes, some of which overlap with patients who have Noonan syndrome. And in our lab in Manchester, we used to do much of the UK's testing for Costello and CFC. So this is now quite an old slide, but I think it may be exactly what appeared in my thesis, which is now also quite ancient history, thank goodness. But what you can see here is that we had a large cohort of Costello patients. And this makes Costello syndrome look quite common, doesn't it? But actually what this really reflects is that this was 20 years worth of patients collected by Bronwyn Kerr and waiting for the gene to be discovered so that they could be tested. So a lot of these diagnoses were made in 2005, 2006, as soon as the gene change had been identified by Yoko Aoki and her group in Japan. In terms of CFC genes, you can see lots of different versions of the BRAF gene can be associated with CFC, but there's this little kind of hotspot, if you like, that is at codon 257, and I think there will be people here at the conference who've got changes at that particular residue. So, Costello syndrome in a bit more detail. Here's a picture of Jack Costello, who's a New Zealand paediatrician, who first described the condition back in the 1970s, and this is really quite an early description by the terms of many rare genetic conditions. And the HRAS gene change is nearly always newly in the individual. And as I said before, often there'll be this antenatal history that's a bit non-specific, but of being a big baby with polyhydramnios, the description for excess amniotic fluid. And then low blood sugars soon after birth. Again, that's quite common with very large babies and babies that don't feed very well. So sometimes that isn't the best clue, but the heart differences, particularly cardiomyopathy and pulmonary stenosis, and this chaotic atrial arrhythmia that we see are really very good clues towards there being a disorder of the pathways such as Costello syndrome. And again, some of you will recognise the young lady on the right in the pictures. There's not a particularly good picture of hands there, but the very wrinkly hands again is something that many of you will remember when little ones have arrived. Many babies can look slightly wrinkly, but there is real excess tissue in the hands for many people with Costello syndrome. And the learning disability, short stature and high tumour risk is something that was sort of recognised quite a long time ago. The tumour risk became established when there were two children, both living in the north of England, with a simultaneous diagnosis of embryonal rhabdomyosarcoma, which, as many of you will know, is an extremely rare tumour in the general paediatric population, but actually relatively common in Costello syndrome. So we think about half of people with Costello syndrome have congenital heart disease of some sort, and pulmonary valve stenosis is the commonest, but other valves can be affected, and also you can have septal defects. We don't think that complex congenital heart disease is something that is common, but with the rarity of Costello syndrome, and also the fact that complex congenital heart disease can sometimes be a very severe thing to have wrong with you, there may have been a child with Costello syndrome who had a more severe heart difference that maybe didn't survive. We think cardiomyopathy affects up to 60% of people with Costello at some point, and it's often left-sided hypertrophy, so that's the main forceful pumping chamber of the heart. About 50% of people have this arrhythmia, which is often around about the time of birth and in early infancy. So again, that's quite unusual, and in the neonatal unit, certainly of our local patients in Manchester, I think the most recent two, really this has been the best specific clue, because often the facial features of babies with Costello syndrome are pretty unremarkable. So here we've got the hands and feet that I talked about earlier. These are of older individuals, but you can see the excess skin, and then as people age, the hyperkeratosis, so this thickening up, which again, some of you, perhaps with older family members, Costello syndrome, might have had this as a thing to deal with. The joints can be very hyperextensible, which is what can give you this unusual hand posture, and then the fingers themselves can have and toes can have quite squared off tips. And ulnar deviation just describes that the hand is pointing towards the ulna, which is that bone there, and flexion at the wrists. And again, all of this will be really sort of recognisable to many of you who look after people with this. So how common is Costello syndrome? We did a snapshot assessment of all the people we could ascertain with Costello syndrome in the UK. This is a few years ago now. But actually, at that stage, it was much more limited where I was offering genetic testing, and so we got a better snapshot than we would do today, I think. And we found that half of them were still living, but the highest mortality was in the first year. And so, of the little ones who'd succumbed, many of them, it was actually in the neonatal unit or in the neonatal period that they'd had something that wasn't survivable. And we were able to find that it's nearly 1 in nearly 400,000 babies that are affected, because we can do a definitive test for Costello syndrome. People don't have an HRAS gene change, then they don't have Costello syndrome. And we were able to talk to the other lab in the UK that was doing the testing over those years, and they told us the patients that they diagnosed. So does the exact gene change in HRAS matter for Costello syndrome? It's a slightly difficult question to address because HRAS G12S is much the most common thing. So over 3/4 of people with Costello syndrome have the G12S version of the gene. And so this makes us slightly uncertain as to what the associations might be otherwise. One of the things that comes up recurrently is for people with G12A gene changes, whether there's a higher cancer risk. And that hasn't really been definitively confirmed, although we do know of some patients with G12A who have quite similar features to one another. And so it might be that actually it is a recognisable thing in some patients. There are also some more severe gene changes in HRAS, like G12V and G12D. And these babies often are the ones that don't make it. And so we wrote up a series of little ones, many of whom weren't actually diagnosed until after they passed, because the pattern was of a very, very unwell individual who didn't make it past the first weeks of life. But on the flip side, there are also gene changes like the G13C, which doesn't seem to give nearly such a severe pattern of differences. And so some of these patients, I don't know whether Colin is the fount of all wisdom on lots of things. Do you know of UK people with G13C?
Not off the top of my head.
No, because you.
In Spain there is one.
In Spain there's one. All right. Okay, very good. Because the American groups have felt that there's about 5% of people with Costello syndrome have G13C. So amongst the people that we know here in Europe, we feel that there must be some people who've got this gene change, but my suspicion is that they might actually be more in the Noonan phenotypic cohort because the pattern is that much milder, perhaps. So here's a little one who had a G12V change, and many of your little ones may have been this unwell in the neonatal period, but with the G12V, it's a rare cause of Costello syndrome, but it's a very highly activating mutation. The gene change does something very drastic to the way that the RAS/MAP kinase pathway is working. And we think that's why we see it relatively commonly in tumours, but not very commonly in the whole individual in the pattern of Costello syndrome. So we used to think that there were features that were unique to Costello syndrome, but actually one of the things that comes out is the more you know, the more you realise you weren't quite right to start with. And the neonatal atrial arrhythmia that I mentioned before is something that we see in patients with RAF1-associated Noonan syndrome, not that infrequently. Embryonal rhabdomyosarcoma, whilst much the highest risk is in Costello syndrome, there are actually now recurrent patients with Noonan syndrome due to SOS1 gene changes who've had that. And similarly, the nasal papillomas that many of you will be very familiar with as a hallmark of Costello syndrome as people get into mid-childhood is something that we've actually seen recurrently in some of our patients with other gene changes in the pathway. So if we think about the new presentation of Costello syndrome, the important thing is to confirm the diagnosis, and then really a lot of it is about addressing feeding needs, cardiac needs, and then the abdominal ultrasound screening, which again is most important up to the age of 10. And we think that that's not particularly well proven as a way of detecting rhabdomyosarcomas early, because these tumours are very aggressive and can actually present as incident tumours in between the screenings. But equally, ultrasound is a safe and fairly well tolerated procedure. We recommend a cerebral MRI, and that's something to be done. Essentially, many patients may have had this done before they received their diagnosis. And if they haven't already had it done, it's something to think about. It's often not an urgent thing necessarily, but a baseline can be helpful, because actually later on there can be changes within the posterior fossa, so the base of the brain, and can enlarge, and actually there are some people within our community who've needed surgery to try and decompress around their cerebellum. And clearly, as the diagnosis is made, support with information is important. And that's something that Colin's done fantastic work over the years in getting out there and getting people on board as soon as there's a new diagnosis made. And one of the highlights of this year really has been the publishing of the new management guidelines by an international group, largely American focused, but actually it's very sensible, and so would be a useful resource for those of you looking after people with Costello syndrome. In terms of the ongoing issues through childhood and beyond, feeding and developmental needs, often feeding does improve by the time children are mid-childhood, and often they will start to be enthusiastic about food even, which is good. Cardiac follow-up, again, it depends on what's been identified, but even for people with no identified problem, we would recommend infrequent follow-up. Abdominal ultrasounds we've talked about, and then a urine test from the age of 10 each year, just to check there's no blood present in the urine, because that can be an early sign that there might be bladder papillomas or an early bladder cancer developing. Again, we know of those being something that are relatively frequent in early years, which is again uncommon in the general population, but they are normally very readily treatable. It's also important to have surveillance for the orthopaedic complications. So many people will get tight Achilles tendons, they can have curvature of the spine, and other things that mean that actually seeing an orthopaedic surgeon or a neurosurgeon is a good idea. And clearly growth monitoring, as for any child with a medical issue, is something that we would imagine would be happening. In terms of CFC, there's quite a lot of overlap. I've already talked about BRAF being the most commonly altered gene in that condition. And the presentation can be indistinguishable from the presentation of both Costello and Noonan syndrome at birth, with congenital heart disease and cardiomyopathy and poor feeding. With CSC, the feeding differences are definitely more variable than for Costello, so there are some babies who do well initially and then develop a problem a bit later. And similarly, there are other children who need PEG feeding beyond the age of 10, which is fairly unusual for Costello syndrome. And the learning differences, again, can be quite significant, but also epilepsy is something that we see quite commonly as a big thing in CFC syndrome, and sometimes having bad epilepsy is associated with having more in the way of neurological difficulties. And those of you who look after people with CFC syndrome will know that it stands for cardiofaciocutaneous syndrome. Sometimes that's a really good, easy description to remember, because people have got a heart difference, they've got a facial appearance that's like other people with the condition, and they've got lots of skin differences or hair differences. But actually we know of people with normal hearts who have CFC, we know of people who look very unremarkable who have CFC, and we know of people with no skin problems who have CFC. And that, I think, makes it really quite a difficult thing for some families to get their heads around, and indeed when actually the main problem is a severe epilepsy, for example, and we do know of patients where that's really much the main issue. There's an argument as to whether this is actually the right name to have given this condition, but again, it works for many people. And does which genes altered in someone with CFC make a difference? The answer is that it can do. So again, trying to assess for differences is tricky because BRAF is so much more common an altered gene than the others. But even exactly which variant it is in the gene is important. And we do see that with our cohort of patients where we know of multiple people with the same gene change who might have things in common. But there's still loads of variability. And so if we think of the Q257R patients who've got BRAF gene changes, some of them have got mild to moderate learning difficulty, others have got much more severe learning differences. And so there must be other factors, which might be other genetic factors, might be environmental factors, might be things about the health, or it might just be total chance that we just can't understand at the moment. So how common is CFC? We don't know, but we do know that it's significantly more common than Costello syndrome. It's difficult for us to get exact numbers because there are some people with CFC syndrome who don't have a genetic diagnosis yet. There are some people where there's an argument as to whether they have CFC or Noonan syndrome because of the overlap in the genes. And there's also a number of patients who probably have got this condition but not necessarily had a genetic test yet. I think that number gets smaller with each year that goes by. And we would certainly hope that most of the people with a significant issue because of their CFC would have had a test considered by now. But actually the people who are perhaps living in supported settings where it's just how they are, those people in their 30s and 40s, for example, may well have been too old to be under paediatric care when the genes for all of these conditions were identified. But we think it's at least twice as common as Costello syndrome, and so we think, perhaps it's 1 in 200,000 people, might be more frequent than that. So here we have pictures. Some of you will recognise these friends of ours. Each of these people have a change in BRAF, and Q257R is the commonest alteration and often gives a fairly significant developmental difference. Feeding problems, really very common indeed. So most of these patients with BRAF gene changes have needed PEG feeding, I think, except for perhaps for some of those with the more unusual BRAF gene changes. So most of our patients with Q257R have proceeded to PEG tubes, I think. And you can see that they can have a big head, and this exuberantly curly hair seems to be very frequent in people with BRAF gene changes, and the skin features of dry skin and the relatively missing eyebrows or sparse eyebrows. Another gene for CFC, some more friends of ours, is called MAP2K1, also called MEK1, and this again is a gene with one variant that seems to be much the most common. And these patients can have a severe developmental difference, but not universally, and there have been a few who've had significant epilepsy. And the other thing that we've noticed in our cohort is that as people with this particular gene change get older, they seem to get stiffening up of their joints. And again, that's a little like we see in some people with Costello syndrome, but actually more generalised. And we don't think this is that common in other people with CFC unless they've been quite immobile because of being very unwell, for example. But a lot of these patients have got fewer skin problems, and also the incidence of heart things seems to be a bit less as well. So that's where we've got the cardio and the cutaneous seeming slightly less appropriate perhaps, and facially some of these people have got very recognisable patterns, others less so. MAP2K2 variants are a rare cause of CFC, and this is more often associated with a milder learning difference, but quite a lot of the ectodermal features, and so again you can see here very curly hair is characteristic, but there are now several families reported in the literature where important MAP2K2 variants have been passed down generations. This is a UK family with a mum and two sons who had a difference, And this was an American family where it was through 4 generations that they could track the gene change. And all of these people had a degree of short stature, a bit of learning difference, but actually they were living very much normal lives and clearly had families and health was not too much of a problem for them. KRAS gene changes are pretty unusual. We've got small numbers of patients and some of them have been the ones that haven't necessarily survived early childhood. And so whilst having a big head is really common in CFC syndrome, some patients with KRAS gene changes in particular can have a smaller head. And one thing that we know is relatively common in this group seems to be this fusion of the bones of the skull, so craniosynostosis. But whilst this is often associated with relatively significant differences, there are some other less usual gene changes that have been passed down through the generations, as is also the case for HRAS gene changes as well, that I'm not touching on in great detail today because there's an argument as to whether they have Costello syndrome or not when there are these mild HRAS gene changes that can be passed through the generations. So another thing that comes into the CFC kind of umbrella is this condition described by Laura Mazzanti in Rome as Noonan-like syndrome with loose Amazon hair, which is not a very snappy title, but we know of several people who had a clinical diagnosis of CFC and actually have changes in this gene. So it seems to sit quite well within the CFC type umbrella, although often the learning differences that people with changes in this gene will have are relatively modest. We know that short stature and even frank growth hormone deficiency is quite common as well. But it's over the last it's nearly 10 years, I think, since we've known about this gene change being important. And there's also another gene that makes a protein that forms a complex with SHOC2 that can, when that protein is altered, it can also cause the same kind of pattern. So we found quite a lot of people with SHOC2 gene changes in our CFC cohort, and therefore that made us think it was worth testing in anybody who's got features of Costello, CFC, or a more significant Noonan syndrome type presentation. So in terms of managing CFC, the recommendations are quite similar to Costello. There aren't any good English language guidelines as far as I'm aware for CFC, and what we've tended to use in the UK is the Noonan syndrome guidelines that we're in the process of updating, and perhaps from that updating process we'll decide if there's anything additional we would suggest from a CFC perspective. The French, I think, have a very well-organised system of bundles of care, really, of what can be funded for people with particular conditions, and they have some CFC guidelines, but rather than being focused on what's common and what should be thought of in all patients with the condition, they have to try and think of any potential complication and then get it into their, it's called a PNDS, I think, Lisa, isn't it? Is that right? So it has to be a much broader thing with many things that won't actually be relevant for many individuals. Is that right? But we know cardiac assessment and follow-up is important, and also cerebral MRI. There are people with CFC who've had hydrocephalus relatively frequently, and sometimes there can be other differences. And I think the brain differences in CFC are not very well characterised as yet. We know that they happen. We know that brains can just be put together in a slightly non-standard way, and especially in patients who've got significant epilepsy, there are a lot of patients now having these high Tesla MRI scans and finding all sorts of subtle differences that we think probably relate to the overall CFC pattern. And clearly support group information, so for the UK Costello and CFC group and for CFC International is something that we provide. And I have to say, when I see families in clinic, I do signpost them to the Noonan Syndrome Association resources as well, because that, again, I think is a very comprehensive organisation, and they've got some quite good stuff on there. So the feeding needs, as I said, can be more prolonged than is the case for Costello syndrome, and surveillance for growth and many neurological or orthopaedic complications is important for CFC. But actually, on the whole, unless people have got severe epilepsy, often their health is less problematic than is the case with people with Costello syndrome. And again, I think with the very variable nature of CFC, making sure you've got a good go-to paediatrician as your point of contact is important in childhood. And I think then there's more of a question as to what happens in adulthood and who the right people are. So in summary, Costello and CFC can present in very similar ways. Costello is associated with a higher chance of specific complications that warrant extra medical checks. And for CFC, we think overall there's probably better survival than there is for Costello syndrome, but it's a more variable condition. And I guess that's something that those of you who are veterans of many conferences will perhaps have noticed over the years meeting people. So I think I was meant to spend a minute talking about feedback from the Baltimore conferences, wasn't I?
If you can, yes.
But seeing Lisa's in the audience, it was an absolutely fantastic conference. It was beautifully organised. And so, as any of you who have been to these sorts of meetings will know, the Costello Syndrome Family Network bolt their conference on to a scientific retinopathy symposium. And so at the CSFN meeting, there were lots and lots of families. I can't remember the numbers.
50?
Oh, 55 or something, wasn't it? It was
I think about 50.
50. And there was lots of excitement about the new management guidelines, because I think especially in the US, where care is absolutely exemplary in some places and much more patchy in others, having a list of guidelines is really helpful for families to then lobby for what they need for their children. There were some talks about skin management, genetics, cancer risk, and those were good, but again, similar to those that we've seen in other years. And the other thing that I think there was quite a lot of discussion about was thinking about older age and Costello syndrome. I think there's been some discussion in the kind of fora about that since as well, hasn't there?
There's been a little bit, but it's still unsure of how things are going to progress.
Yeah, and so I think that's, again, whilst the adult phenotype of Costello syndrome has been quite well set out, and there's a recent paper by Nicole Weaver and others about that, perhaps looking at people who are 18 plus is going to be different to looking at people who are, I don't know what's old, 41, for example, plucking a number out of the air. You know, people who are 40 plus might have a different pattern of needs, for example. And so in the scientific symposium, there were some good illustrations of the work that's going on to understand the biology of Costello syndrome and CFC and related MAP-kinase pathway disorders. There were some really nice presentations of new mouse models of these conditions, particularly the Costello mouse that's been developed in Japan, and whether that's got some metabolic differences. Sounds like there's lots of potential for them to develop that further. And then also, I think one of the messages for me that came out of it was thinking about, we do have agents that will target the MAP-kinase pathway, that will stop it from working quite so much in overdrive, but actually when you use those in a biological system, like an animal model or a cell model or a human in due course, it doesn't always work as well as you'd hope, and that's because of the complexities of these networks of signalling. And actually, what they did have was some really nice emerging data about combining drugs to limit the amount of toxicity that people get, but also actually be far more effective than the single agents. And again, I think that's something that hopefully in 2 years' time there'll be more to think about. So I'll just stop with an acknowledgement slide. That's quite an old picture now. I think it was when you guys all went to London Zoo, probably.
Yeah, 2010.
Clearly Bronwyn is who I've learned most from over the years, but all of you as well. So thank you.